Hello, welcome to Examining Evolution. My name is Rebekah Davis, and I'm here with Sal Cordova. Sal is a molecular biophysics researcher, and he's been on the channel several times. Sal and I share a mutual skepticism when it comes to evolution, and we kind of feel that mostly what we hear in the popular media is just, that the evidence for evolution is overwhelming, and that this is kind of just we've got so much evidence for it, but we don't often hear the actual problems, the incredible obstacles to this idea of organisms evolving from what I'd say is like amoeba to antelope, right? Where you've got to have some sort of process to get things from basic to more complex. And so tonight, Sal and I wanted to talk about what one of the issues, one of the very difficult problems with evolution. So we're going to be talking about evolution of eukaryotic components. Sal, would you tell us a little bit about your work and about what we're going to talk about tonight?
Yes, I'm a molecular biophysics researcher, independent. I'd worked for world famous genetic engineer, Dr. John C. Sanford, retired research professor at Cornell, and his invention, the gene gun, is in the Smithsonian National Museum of American History, and he became an evolution skeptic after accepting it most of his life. And so let's just talk about a little bit about what I know I'm jumping a little bit ahead. We're going to assume common ancestry is true, but we're going to criticize the difficulty of the complex machinery emerging. And that's what a lot of my work had been studying, these complex machines. I'm published. I'm very grateful to be published and in a Springer Nature reference work. It's on university library shelves now, and it happened this year.
Congratulations.
Book retails for $1,500. I don't get a dime if you buy it. I know I have a fan club out there. Just wanted to let you guys know. There are ways to get a hold of our reference chapter. And I also work in the area of topoisomerase and protein fold prediction for medical science. And it's also given a chance to criticize evolutionary theory through that. The publication that I mentioned, the Springer Nature reference work, was actually critical of evolutionary theory. But the way we got it past peer review is we just simply quoted top-tier evolutionary biologists criticizing their own field. And so when we examine problems with evolution, we're going to examine it from a purely academic standpoint and give you aspects of it that even the academics themselves, who are pioneers of this field, when they have an honest moment, they publish it in peer review, they use technical language, and they really admit all the problems. It doesn't get a lot of press, and a lot of people don't know it, even other evolutionary biologists, but I've dug some of this stuff up and I'm happy to share it. And so, so there's kind of my critique of evolution work, but then also the medical work is really exciting. We're having new frontiers in using bioinformatics and biophysics and computation to just figure out just the intricacies of these molecular machines. So we published there. My specialty has been in topoisomerases. I also like proteins like collagen and zinc finger proteins. So I know that was a mouthful of terms, and there's a lot to learn about biology. It's, the Earth's biosphere is probably the most complex system in the entire universe. The complexity of machines here we don't find anywhere in the universe, and I don't expect that we will.
Cool. Well, do you want me to bring up your slides? Or how do you want to start?
Okay, so we're just going to start with the basics and go ahead. Yes. So there are two ways that there are a variety of ways to kind of classify major groups of organisms. We're finding that they're each of them share enough that it's a little bit somewhat but not completely arbitrary how we group them together, because there's certain features that really stand out. So we say that there are basically two empires of life. If you look at the Wikipedia entry on two empires of life, they're eukaryotes and prokaryotes. And within the prokaryotes, you have bacteria and archaea. And then in eukaryotes, sometimes we just lump them all together as eukaryotes. But in the Wikipedia entry, you could see how it even further subdivides it into plants, the plantae, the animals, the fungi, and the protista. So that's just kind of the division. Now, what we're finding out is that the way we used to define what makes a eukaryote, we'll say, well, certain things only exist in a eukaryote, but not in the prokaryote. And it's more fair to say that it's vastly overrepresented in eukaryotes, and maybe only a few bacteria have that particular component. So maybe, I've been thinking about this and say, that the two are really distinct, but not in a absolute cut and dry matter. They're distinct in that certain components, like eukaryotic components, are very much represented in eukaryotes, and maybe we find only a very, very small percentage of bacteria have that eukaryotic like feature, and same for archaea. So we'll cover what those components are. So people say, well, evolution's just proven beyond a shadow of a doubt. And I'll say, well, first thing is, what do you mean by evolution? If you mean common descent, I'll say, okay, I'm going to grant you common descent. I'm going to grant you that common descent is established. People who know me know that I don't believe that, but I'm just like, let's just teach this from a mainstream textbook standpoint. Say, okay, let's accept the mainstream view that common descent is real, just for the sake of argument. And I know there's some people that don't hold that, but let's just do that as a as an intellectual starting point. If we assume this, does it naturally proceed that these eukaryotic components would emerge? So the standard theory is, and there are many, this is mostly non-controversial, but there are exceptions. But let's just go with the mainstream theory that some prokaryote existed first, and that was the simplest life form. We would assume that it would be a prokaryote-like creature. And then from that, a eukaryotic-like creature emerged. The problem is if we just assume that pathway going from, okay, I'm glad you could see my cursor here, going from a prokaryote to eukaryote, we would need such radical changes. The changes would seem indistinguishable from miracles. And the evolutionary biologists will say, no, it was a gradual change. It was a gradual change. You don't understand evolution. And I'll counter, and I say, okay, so if I don't understand evolution, why don't you explain how these components could evolve in a stepwise fashion? And they're never able to explain it. There are things that you can explain that evolve in terms of a stepwise fashion. A very easy, you could take down the picture briefly here, Rebekah.
Okay.
So let's look at some things that evolve in a stepwise fashion. How about height and weight? We could see populations of individuals, be they human or something else, just change in terms of size gradually. That's stepwise fashion. It's totally easy to see that. That's not controversial. And then when you look at the fossil record, as far as like human evolution, and we see kind of primates and kind of the mammalian stuff in the fossil record, it's like, yeah, you could kind of see little stepwise evolution there. And even though I reject common ancestry, it's like, I'll grant that. I totally see that. The difference is when you get down to the molecular and cellular level, the leaps are just gigantic. You're just like, no, this is just for me. What happened to me is like, yeah, I'll grant that, size and shape can move in a stepwise fashion, but not these molecular systems. This is like differences between passwords sometimes, or like locks and keys. If it's just like, okay, how do you evolve a blender into a piano, or how do you evolve a piano into a car, or a car into a submarine? It's like some of the stepwise things just kind of beggar the imagination. So in the human world, where the kind of like what we call say like a transitional form, it just doesn't make sense to evolve one thing to another. And I'm running short of analogies here. If you have any questions, that's where we're headed.
Cool. Do you want me to put your slides back up?
Yes. Okay. So let's look at some of the differences between eukaryotes and prokaryotes. And we've covered this on other discussions, but just to review, the main thing that separates eukaryotes from prokaryotes, and again, there are going to be slight exceptions, is these membrane-bound organelles in eukaryotes. So membrane is made of lipids. So fats are a form of lipids, just to give you an idea. I'm going to throw a lot of terminology here, but that's just kind of the nature of the field. We just have to learn terms that are alien to our everyday life. So on the left there is a prokaryote, and a prokaryote, you can see it has this nucleoid. The nucleoid, that stringy thing in there, is the DNA. Whereas in a eukaryote, the DNA is kind of in here in the nucleus. But it is enclosed in this thing called the nuclear membrane. And what the nuclear membrane does is it prevents things from going in and out randomly. It has to be a very controlled gateway for stuff to go in and out. Whereas here, it has a lot more freedom of access. And then you have all these other organelles, inside a eukaryotic cell, and they're also membrane-bound organelles. These membranes are an enclosure, and they have gates that let things go in and out. And I've criticized evolutionary theory. I said, okay, let's just assume common descent is true. The problem is, if you don't have those gates in place to begin with, the system or possibly the whole creature is just going to be dead from the start. It makes no sense to make an intermediate. It's just like saying you have an intermediate password and you're trying to log in. It's either all or nothing. A lot of these systems are all or nothing. And so if you have any questions, that's kind of one of the basic differences, but there are plenty more if you have any more questions.
Okay, well, I guess my question is, because if I'm just looking at the shape of the prokaryote and the shape of the eukaryote, I might imagine the prokaryote just getting a lot of stuff added, and like what I see in the middle of the eukaryote just somehow, grows around, like it just grows more stuff.
Right.
So my question is the cell wall so different from the nuclear membrane? Like, is there a possible transition there? Like,
it's actually very similar in terms of the wall. What makes the difference is the little gates.
Okay.
The gates. And this is the amazing thing, is when stuff goes in and out of the gates, and you could see that it has to go elsewhere in the cell, and then it has to go back through another set of gates. So you need like multiple passwords, so to speak, to go in and out. And that is, so one can imagine it, and that's fine. The problem is when you get into the details. And the way we sort of know that it's problematic is when we shut down these gates, the cell dies or the system dies. And usually this is how we find out how stuff works. We just kind of try to break it and see how important it is. And you break it, it's like, oh, that was pretty critical. And that's how we can start to kind of make testable claims that this is difficult to evolve. So, let's compare that again to evolving size of a population. You can make something a little smaller or bigger and it doesn't necessarily die. It's very different when you try to do stuff like this. You either have a cell, a membrane wall with or without gates.
Well, now you're saying gates, but gates don't sound very hard to me. Like, to me, that just sounds like something needs to open and close. So is that really hard? Couldn't you just like get a flap?
Well, the thing is, you have to have a recognition system. That's a great question. You have to have a recognition system. So just like if you have a doorkeeper and he wants to know whether you're really allowed or not, he's gonna check you out and ask you to identify yourself. That identification system is really important. And I've shown videos on nuclear import-export. I don't know if you remember any of those, but everyone who watched that they're like, that's kind of jaw-dropping, the complexity of these mechanical gatekeepers inspecting the cargo, so to speak, and saying, hey, you can go in here. And by the way, some of these cargoes have to be enclosed in things called importins and exportins. So it's more than just the gate. The importin and exportin, kind of like their trucks, so to speak. The truck has its own gatekeeper and say, hey, I can transport you. Do you have a bus pass? And if you do, hey, hop on board. If not, don't. And to organize that for like even say a life critical protein like say topoisomerase, because that happens to be one that I study. So you have to have the truck, you have to have a topoisomerase that has its bus pass. Then it gets into this an importin, and the truck driver, so to speak, of the importin says, hey, hop on board and I'll shuttle you through the gateway. So then the importin has to go through the gate of that nuclear pore complex, and the gate has a guard called the nuclear pore complex. It's assembled with like 300 proteins. 300 proteins! And, oh, Rebekah, you're muted.
Sorry. So this isn't just a simple thing that it can change. We're talking about 300 protein change, and even every one of those proteins is quite complex in itself.
Yes. Not to mention we have to have machinery to stitch the proteins into the cell wall. So this is like building a cell wall, and then you drill the hole. Like if you've ever seen how houses are constructed, or
Mm-hmm.
You have to build the hole. You either build the wall and then you drill holes, or you have to kind of build it with a hole. But basically, you have to have a mechanism to stitch, like your window or your door into the house. I mean, it's not a trivial thing to install a door, and so you need a host of machinery just to do the installation of other machines. So you need a host of machinery to build the wall, and then to build the doors or gates. And then the gates themselves are machines. And then the gates have all these kind of, all these these little entry codes. And then, you have the trucks, passing through the gates. And then the trucks are inspecting for bus passes of the proteins. I mean, and they're like, oh, the evolutionary biologists will say, oh, no, you're not accounting for natural selection and how this can be built up incrementally. And I'll say, okay, fair enough. Why don't you describe it? I'm just giving you all the mechanical details. And all of them said, we don't know how this could come about. And so I'll press them and I'll say, well, to me, this looks really improbable. You have faith that there's an answer in terms of ordinary mechanisms, and that's fine. But you're admitting this is a faith belief, not one that you've actually demonstrated. So if you're going to advertise that evolutionary theory is well established, and yet on the other hand, you're telling me these big, gigantic problems are not solved, I said, there's a little disconnect here. Don't you see? So I already said common descent's okay but the problem is we keep changing terms on the fly. What do you mean by evolution? Do you mean common descent, or do you mean the emergence of complexity? And a scientist like Michael Behe made that distinction. He believes in common descent, but he says the emergence of complexity, he's the one who coined the word irreducible complexity. He said that's the real problem. It's called the biochemical challenge.
Now, we have a comment in the chat, a question in the chat related to, so Nidimo says, it seems there's already working theories on this problem that we're talking about, I guess, with the gate.
They're always working theories, and they're based on, I've read them, and I'll make a testable prediction. They're going to be the same recycled hand-wavy arguments that aren't really worthy of a scientific hypothesis, meaning it's circular reasoning, and they're asserting that these things evolve naturally, and that's all that these papers say. Oh, this thing had to evolve before that, but they never get down into the mechanical details of the probabilities of estimating how probable it is that you'll get the right mutation, and how probable is it that you'll have a selection pressure to make it. If something is dead, there's no selection pressure. That's the problem. That's the problem. What we call natural selection, and I think that's actually a terrible word. It's more, the better term, more accurate, is differential reproductive abundance. It will not favor a half-formed system, because a dead system will just not reproduce. And so there's not going to be any gradual pathway to this. And they'll say, well, the way it is now wasn't the way it is then. And I'll say, fine, you can imagine all you'd like to propose, but at least make an imagination that is mechanically feasible, which they never do. And just admit you're accepting this on faith.
Your buddy, Dr. Joel, is here.
Yeah, see, Dr. Joel, we attract all sorts of I told you.
All right.
We're attracting all sorts of high caliber people here. And Nadimos just said, what about endosymbiotic theory? Endosymbiotic theory will not solve the problem of the nuclear pore complex. So again,
what is endosymbiotic theory?
That there was supposedly a eukaryotic ancestor that ate up, for lack of a better word, the proper term is engulfed, a bacterium. And that bacterium ended up being the mitochondrial. So if you show my slide again, it's saying the eukaryote evolved from an ancestor eukaryote that didn't have this mitochondria. And then so this prokaryote got engulfed and then incorporated into the eukaryotic cell. And I'm going to even grant that as partially true.
Okay.
It doesn't explain. The problem is it only would explain at best this. It wouldn't explain this. So that's the problem. The other thing.
Now, are you granting it as partially true because you think it's partially true, or just because you don't want to argue about it?
Because I don't want to argue about it, but I actually do want to argue about it, but it would take too long. But while we're there, let me just point this out. 85% of the proteins that make this mitochondrion have to come from here. And a lot of people will say, we did this phylogenetic reconstruction. It shows that absolutely came from an alpha protobacteria. And I said, then I'll cite, Penny, Collins, and Kurland, and said, well, 85% of what makes this has to come from genes that are in here. How'd that happen? And then you have to have the genes coming out of here, and again, going through the nuclear pore complex, then getting coded somewhere, and they have to go in here. So you have basically two gatekeepers, and then you have to have a transport process going from here to here. And how, if there's anything in here that's life critical to here, it's like, how could there be an intermediate? So there are problems with endosymbiotic theory, but I'll even grant that despite all the objections I just put forward, it will not solve the emergence of the nuclear pore complex here. So, and we're not even covering some of the other things in eukaryotic components. So I'm glad this is a great review.
Okay.
But I haven't even brought out the heavy artillery yet.
Okay, well, let's move on.
Okay, so I was in a debate with Dr. Daniel Stern Cardinal. He's a evolutionary biologist and professor of biology at Rutgers University. I consider him a distinguished colleague, and we absolutely do not agree on evolution, but we're able to get along. And one way we do that is we just talk about the architecture of biology. So let's just talk about the architecture of biology, things we can agree on. And so we have this is a picture of what we call the central dogma of molecular biology. So you're going to learn the heart of molecular biology is right here. Dr. Change Tan teaches molecular biology, and she'll start off saying, okay, this is the basis of what we're going to study in molecular biology, the central dogma. So the idea is you have DNA. DNA transcribes to RNA, and there are machines that take the DNA, make an RNA transcript, and then the RNA transcript is used to make a protein. Now, there's some subtleties as far as RNA can be reverse transcribed into DNA, and then there's all sorts of things with the replication cycle of DNA. But basically, DNA makes RNA, makes protein. You can't go the other way. You can't take a protein and make an RNA that goes into a DNA for the most part, as far as I know. And this is textbook. So do you have any questions here in this?
Well, I'm curious about what Dr. Joel is saying here. He says horizontal gene transfer from empty genome into the nuclear genome has happened within kinds very recently. Can you respond to that?
No, thank you, Dr. Joel, for that information. Feel free to drop in the chat how that was established. Was that done by actually observing a cellular process? Were we able to actually observe the DNA transfer from the mitochondrial genome to the nuclear genome in vitro under controlled laboratory conditions? So thank you for that. So that would raise the question then how, there could be an established transport process there. So thank you for the data point. Much appreciated. This is great. We're actually getting critiques and editorial feedback on things that I claim. And if I'm wrong, I have a responsibility to make sure that I deliver the very best and cutting edge stuff to the listeners. I will point out that Dr. James Carter, who's a biochemist and professor of biochemistry, and he teaches molecular biology type stuff, he shares my view. So this is a great discussion to have. We're not aware of any of that. But nonetheless, even if that were true, hypothetically, that wouldn't negate anything I said about the nuclear pore complex and the complexity of the 300 proteins, and then plus all the host of machinery needed to build it and to implement it. So anyway, but thank you for your comment, Dr. Joel. On an exam, I'm sorry.
I'm sorry.
Yeah, on an exam, if you're taking molecular biology, they probably want you to recapitulate this diagram. But informally, we could say DNA makes RNA, makes protein.
Okay.
And so when they say DNA is the blueprint of life, it's more accurate to say it's the blueprint of the RNA, which makes proteins. Or you could skip and say DNA is the blueprint of proteins. There are things we're finding out that we inherit that are outside of DNA, like membrane-bound organelles and glycans, and we don't need to go there. So it's not. We're learning that it's not exactly right to say DNA is the blueprint of life. It is the blueprint of the proteins of life.
Okay.
All right. So this is where we get the concept of genes. So there's a stretch of DNA that will make a particular protein. So a gene corresponds to the protein it makes. A gene is the blueprint for a particular protein. Now that's like in biology, it's the science of exceptions. So we'll just move on. But the difference, let me show you the difference. If we take molecular biology, even some more, the way that the prokaryotic gene makes a protein is different than a eukaryotic gene. So you could see this thing called the coding region. So we've shown a lot of movies of like the topoisomerase.
Mm-hmm.
So let's just imagine in a bacteria, the topoisomerase is just coded by one gene. It's actually coded by two in the bacteria.
Mm-hmm.
It's called the gyrase. But just for simplicity, let's say hypothetically we have this hypothetical protein and it just has this coding region. So the DNA here ends up being the blueprint for that protein. And then we have all these other parts of the DNA that aren't coding. In the eukaryote, it's very different. We have this thing called an exon, and basically you have the blueprint kind of just sliced up and had little spacers called introns inserted.
Mm-hmm.
And then you can make a coding region from it.
Oh, wow. Dr. Joel added some information in here. He says inference, but a strong inference from genomic data. But he says he agrees with you about the other aspects of the eukaryotic genome, just pointing out that genes can be moved from one organelle to another. There are very large fragments of the mitochondrial DNA genome. Anyway, so
in the CP found in the nuclear genomes of species, in some cases, the entire mtDNA can be found inserted. And that assumes common descent and that there was some mechanism. And I would, my only comment is let's just assume it for the sake of argument. It'd be worth examining the difficulty of implementing all the machinery to make that happen. Just like we can say, hey, there are nuclear genes that go in the mitochondria, we'll just assume it evolved. What I'm attacking is the mechanical feasibility of this and trying to put probability stuff. So thank you very much for your comment.
Okay,
so,
I see.
For the most part, I'm just going to assume, let's say it happened, but I'm going to assert, I think the mechanical feasibility is a lot harder than we're making it out to be. You can assert that, but then we critically examine the evolution of it in terms of mechanical details. We'll be like Michael Behe. We'll grant all that, but let's see if the probabilities line up. And if it's very improbable, then you're confronted with, how much can we assert that this is natural, ordinary. Natural is a metaphysically loaded word, so I'll just say ordinary or extraordinary. All right, so let's get back to here.
Okay.
So you could see that this is actually a big problem in evolutionary theory. Why would we have genes? Okay, let's say we evolved from a prokaryote to a eukaryote. Why all of a sudden are you putting little spacers in here called introns and chopping up the gene? What you need to have then is a massive machine to undo it, to reconstitute it. And that's called, let me see if I have a picture of it. Well, here's another picture of it. So you have this thing called exons and introns. And
so you need a machine to chop it up.
And this is the machine called the splice. This is just a massive process. It's not, you have this gigantic machine here called the spliceosome. It's one of the biggest machines. And the, I think the subtitle of this was the machine of the spliceosome. And I know someone really hates the word. Oh my goodness. The term was the design of the spliceosome, the design of the spliceosomal machine. And this was in a peer-reviewed article. And we know one of my detractors just hates me using the word designed, a designed machine, but that was the title because that's what it looks like. This spliceosome here is going to go here. The DNA is going to make an RNA transcript just like here. So you got DNA, RNA, and that's called raw we'll call it raw RNA. A molecular biologist will get on my case if I'm using kind of weak terminology, but it takes that RNA, and then the spliceosome is going to come along, and it's going to chop the RNA. It's going to chop out the intronic RNAs, and then stitch the exons together. And this machine is like at least 100 proteins.
Wow.
What happens, just kind of like with the topoisomerase, you saw it cutting the DNA, chopping it. This one's really chopping the RNAs to smithereens and restitching it together. What happens if that spliceosome is not doing its job the right way? You're going to have a disaster.
And you need 100 proteins to make the spliceosome. So, and each one of those has to evolve by a step-by-step process.
Right.
So,
Right.
and they've got to come together all together properly in order to do this function.
Right. Now, bacteria have some way of stitching to get, they have what they call some sort of introns that are self-splicing. And I know some of my detractors will point that out, and I'll say, okay, I'll grant you that. But where do you come up with all those 100 proteins? I mean, why would differential reproductive abundance, also known as natural selection, although that's a misnomer, natural selection, why would it favor just making a gene and chopping it up like this? What good is that? And then you have to make this even more complex machine that's going to undo it, basically.
And how do you know you need the machine? That's the problem.
Exactly.
You need somebody needs to, have like an awareness, right, of what needs to happen if you want to splice it up. So
Dr. Joel had a very nice compliment. Thank you, Dr. Joel. He said you're totally right. Totally fine terminology, Sal. So thank you. That's very kind of you, Dr. Joel.
So
Yeah, go ahead. Yeah.
No, look, I have to just say something here because, guys, I just really hope this is a wake-up call for people who are watching this. Do you realize this is only one little part of the cell, and we're already need so much to happen for it to function? And like when Dr. Joel said something about the probabilities. Yeah, you've got 1 trillion opportunities, or 1 in a quadrillion. Okay, but I mean, we need these to be organized. It's not just having the opportunities, but I mean, I don't care if you have 100 trillion, but they've got to organize together. I really hope people can just see this. Sal, you're doing a great job. Thank you so much.
Okay, let's just zoom in a little bit here. Okay, I want you to appreciate oh, sorry, totally messed that up.
It's okay.
So you have the DNA here, and let me see if I can I grab, okay, so you have the DNA here. And remember, in a eukaryote, it's in a chromosome, and then it's also wound up in something called histones into nucleosome complexes. And that's the next part if we ever get to it. But let's just keep plowing on. So this DNA, then, you have all this machinery that's going to transcribe it. And that's what they have, this thing called polymerase 2. It's a machine that's going to make, it's going to read the DNA and then make an RNA transcript. So this RNA transcript has all this stuff with introns and exons. The corresponding RNA has introns and exons. And it goes into the spliceosome, and the spliceosome is going to just chop it up and throw out the introns. Okay, so the introns will be gone, and that waste product, quote unquote, has to be processed. It's actually not waste. We're finding out what it does. And then it stitches together the exons together, and then it comes out here, and you get this pre-mRNA. The pre-mRNA then gets all sorts of additional processing. It puts on this poly-A tail to it, and that gives it all these codes. These codes enable it then to pass through the nuclear pore complex. So remember I said all the gatekeepers?
Wow.
So here it is. You're splicing and dicing and repackaging. You're adding even more, like you're adding a postal address here in the poly A tail. and then that tells it to go out here. So now this new mRNA transcript goes out here, and there are all these machines that are going to translate it into a proteins, these ribosomes. And sometimes you have these polyribosomes or whatever. It's just really complex. And then, so now you have all these proteins floating around. And then the machinery has to decide, the transport machinery says that the protein go back. Like in the case of topoisomerase, it has to go back. The protein has to go back. So it rides this importin truck. Remember, it has to have a bus pass. And then, so the bus driver, the importin, says, hey, you can ride with me, and I'll shuttle you back through the nuclear pore complex. The nuclear pore complex will check whether that bus is allowed to go through, and then he'll go back in here, and then you saw how the topoisomerase will do its thing. And the thing is this, remember, the spliceosome is made of proteins. The spliceosome itself, all the components have to come from the DNA. The DNA is going to have to go through the spliceosome and then go through this process, and then that's how you can make another spliceosome. So there's a chicken and egg problem here. If you don't have a spliceosome, what are you going to do? If you don't have a spliceosome to start to make another spliceosome, you're stuck. I'm just saying,
you need the gates, you need the spliceosome, and you need the awareness, like something in this process has to be aware that the code is needed for the gate.
Right.
Like, and then the code is needed for getting back through the gate.
Oh, there are codes at every level. The codes at every level. I mean, the DNA has to have in the gene foresight. It has, I mean, if I were trying to put this together, I'm like, okay, it's hard enough as me as an engineer to say, I have to account for all this, because these codes here, when I'm making the DNA gene, I have to have foresight that it's going to go through here, and the introns are not trivial. There have to be codes to small ones to enable the spliceosome to say, hey, I need to chop this out or not. That's not trivial either. So you have to engineer that. Otherwise, you get a mess. But let's say you engineer that. You have foresight. When the RNA transcript comes out here, and then it's translated to a protein, you had to have foresight that you're going to put these called peptide signals, which is the bus pass. And the protein has to be shaped to be able to be loaded onto the, when you're trying to load a truck, you have to make sure that the cargo fits the right way. And the importin truck, so to speak, will allow only certain shapes. And you need the bus pass in the right shape. So you have to anticipate all this if you're designing this from scratch.
Now, what's really frustrating to me about this, Sal, and I really hope people will watch this and think about it, but like, if there was like a building that had like security guards and codes that you had to enter, and like machinery within, machinery without, like no human being, if you said, okay, you've got millions of years for this to develop naturally, no human being that has a brain in their head would say, oh yeah, okay, I think that could happen. Like, we would dismiss that as ridiculous, right? But
there's some really bright guys who do exactly what you say. And, some of them, like Change Laura Tan, who accepted what you just said, that, enough time it'll work. When she started teaching this class, she had to teach her students the difference, what I'm just going through here, this basic molecular biology. She came to the realization, this is really improbable. And it changed her viewpoint about evolution. It's basically what I'm saying is that even let's accept common descent as a principle, you would need miracles, statistical miracles that are indistinguishable. You need statistical events that are indistinguishable from miracles.
Well, yeah, and I guess I shouldn't say no human being would do that, because you're right. They are doing that. There's a lot of smart people that are doing that very thing with what we're looking at right now. They're looking at it and saying it arose by chance. And it's just as ridiculous as saying that, a building with guards and codes and machinery inside and out came together by chance.
So, I mean, let's just...
And natural processes.
So, I mean, it's becoming fashionable now to say, okay, we recognize now the odds, like say in abiogenesis, which is different than what we're talking about. But when they deal with the odds, they say, we've come to terms with the odds. It really is remote. Therefore, the only way to solve this is that we have to assume that there are infinite number of multiple universes. We just happen to be in that lucky one, because they do recognize the odds. And they'll say, hey, we're really just very, very lucky. It can happen naturally, and we're just part of a system of infinite number of universes, and we just happen to be in the one that made it. And I'm like, okay, I will even grant that. We can at least agree that the odds are remote. And what I'm showing here today is not just in abiogenesis, but the evolution of eukaryotic components. And one of the eukaryotic components are the spliceosome is one of them, and the nuclear pore complex. And then all of the machinery that has to stitch it, and all the machinery like the trucks, the importins and exportins, and all the chemical stuff and the fuel factories. Yeah, that's...
Well, now, and Dr. Joel, I think he's responding to what I'm saying here, or maybe he's responding to you, I don't know, but he says, you seem to be assuming randomness, but there is nothing random about the process of how gene regulation and gene functions change over time. Things don't come together by chance. I can agree there.
Hey, we can agree on something. So the question people will say is then a Darwinian process, what he called natural selection, might be able to accumulate the changes over time in incremental steps. And I gave a show yesterday and the day before with Emery Moyna and said, well, the experimental evidence shows that differential reproductive abundance, what we have misnomered as natural selection due to Darwin, actually tends to select for genomic decay. So if we want to say that it's not a random process, the process that we see is actually genome reduction. And we went through 4 hours of this, just citing what evolutionary biologists have said, the experimental evidence, and also kind of the theoretical justification for this. And the reason for that is there are many ways. I call it Lynch's principle, after the evolutionary biologist Michael Lynch. Just like a password, there are many ways to break, only one way to make. For like the sensitivity of a password, there's only one way to do things. We can kind of relax that a little bit and say there might be many ways to make a protein, but relative to the ways that we could, make those polypeptides, it's a very small space. It's a very small fraction. It is a highly improbable fraction. So we could move on, but thank you for your comment.
Yeah, and he also says, of course, it's easy for me to say I don't believe in chance anyway, since I ascribe to full sovereignty of model of creation. What does that mean?
Oh, yes, I would love Dr. Joel to explain that. I think he sounds very much like Sygart. And I saw you in a video. You were at his conference, and you're asking, and he said, Rebekah, and you're asking, how do you account for all these machines? You accept evolutionary theory. He said, yeah, I accept the evolutionary theory. But then you asked that question, and he said, I think there has to be a theistic explanation for this. So he's as mainstream as it gets. But yeah. I was just like I was expecting him to say, oh, we don't know. And he was a professor at my graduate school, by the way. I never took a class from him. So I have the greatest respect. And cool. I was like, wow, Dr. Gart, that was pretty daring. You're going to metaphysical territory here. So there are people like Dr. Gart, and I would just, I'm taking a risk by speaking for Dr. Joel here, but it would seem he and Dr. Gart have,
similar views.
Like-minded thoughts.
Okay.
I'll let Dr. Gart speak about his viewpoint in the side chat or anywhere else, lest I misrepresent him. But thank you for your thoughts here.
Yeah.
So But let's say it is even the sovereign creation thing. What we can say is that if it's not a chance process, it violates what we call in statistics expectation. So it's just like when you enter a casino, and I used to be a professional gambler, you don't expect the dice rolls to be 7 every time. If that happened, it would be a violation of expectation, and you just destroy the casino. So maybe the way to frame it is this would be rather than say, this is non-random. Let me just for your sake also, Rebekah, and the viewer's sake, the proper way to say it is it would be a violation of expectation, a violation of natural expectation. Violations of natural expectation. That's a scientific way of saying it. Saying it's random or non-random gives all sorts of, it's going to invite a lot of arguments. So that probably was good that Dr. Joel challenged me and kind of said, I don't believe in randomness, and so I need to use better language. So the better language is to say it violates expectation. So it violates our expectation. Okay. Rather than say we don't expect random chance to assemble buildings with guards and passes. It's more proper to say it violates natural expectation that it would assemble through such a process.
Yes. Okay.
So now we haven't even, we're giving you, this is, now we're starting to touch on the heavy artillery here.
Okay. All right.
So, we found a bacteria, and I reported it. It was reported only in the last 2 months, I think, that there is a bacteria that does have a nuclear type membrane. It's nuclear-like, but it's a very rare bacteria. It's very atypical. So that's why I call it, when I say it's a eukaryotic component, it's a component that is very well represented in eukaryotes and not in prokaryotes. And so all these guys that are very anal about classification, they're kind of upset. It's like, well, we used to be able to make this hard and fast assertion that no prokaryote will have a membrane-bound organelle or anything that looks like a nucleus, and then we found something. So it's like, okay, I needed to soften my stance on this and say, okay, let's just talk about the components, and then the argument holds. So our mutual acquaintance, who's my detractor, was saying, oh, Sal changed his mind, and yes, I did. But I don't think, in light of what we're seeing here, that it's lessening the basic difficulty of evolving this, because it looks like it violates natural expectation. So I hope you've enjoyed just learning a lot. I've given you like half a semester's worth of molecular biology right here.
Cool. Well, let's keep going.
Okay. Can you... I never would try to interrupt the stream, but I want to just, I wanted to check if I was expecting an emergency call.
Sure.
Yeah.
Well, do you want to wrap it up here? And
no, I want to plow on let's plow on and finish out a little bit more.
Okay.
Because everything I've said so far, you could see, all these little components are worth shows in and of themselves. And we had a show on this. We could have a show on this. We could have a show on this. We could have a show on this. I mean, it's just beautiful. But let's move on. And so in my debate with Dr. Dan, we're talking about junk DNA and non-coding DNA. And I was going to defend the idea that DNA is more than just providing codes. So here's the central dogma of molecular biology here. We look at DNA as being primarily as used to provide a blueprint for proteins. The problem is, in humans, only 2% of the DNA is an actual blueprint for the protein. So they said we have oodles of this non-coding DNA, and we said, well, okay, let's grant some of the other DNA is used for other things that we've experimentally established. We could bring the number up to 15% is actually used for something. 2, that 15% is used to make proteins. But then say 85% of the rest would be considered what they
Junk DNA.
Junk DNA. So so junk DNA is a fraction of all the non-coding DNA. So 98% of the DNA roughly is non-coding, and 15% that contributes to that 98% is, let's say 13% is kind of used for something, but the rest is considered junk. But that's assuming the DNA is used solely for coding and regulatory purposes. So the other parts of the non-coding DNA are for regulatory purposes. And you can why don't I just cover that? So you have here, they're they don't have it in this diagram. They're parts of what they call like promoter regions, etc. And other sort of sequences that kind of give the addressing, to give the machinery that decodes it. It gives it kind of positioning stuff. It gives codes on the DNA that says here, park here, and those are called regulatory regions. And I'm sorry I don't have a diagram for that, but let's just keep plowing on. So they came up with junk DNA, but I said, well, let's look at DNA in a different way. By the way, this guy's Waddington. He created the thing called the idea of epigenetics, and we're going to cover a little bit of that today. I just like the way he looks with that pipe and that jacket. So fashionable. I actually have a jacket like that, and I have a pipe because I just thought he was so cool. So anyway, DNA is not just about blueprints for proteins. It can act as a scaffold. So here's a picture of a scaffold. So when you're building a building, you have ladders and you have this scaffold so that the workmen can come and bring materials and use their tools. And you have gigantic scaffolds like this where you can bring materials and tools and other machinery. So now it changes the way we look at DNA if it's not solely for making proteins, but as a scaffold for molecular machines to help in the regulatory process. And I'm going to show a little bit of that, and then we can wrap it up. So give me maybe 20 minutes.
Okay.
In my debate with Dr. Dan, I got through this in 20 minutes. I don't know how quickly I can get through it here. So I had the picture of the scaffolds, and now think of parking lots also.
Mm-hmm.
So you could see that in the scaffold, you have places where the men can stand, where the machines can be put, and the materials. We can also look at it as parking lots. So we're using two kind of metaphors or analogies.
Mm-hmm.
Look at it as a scaffold or as a parking lot. In the parking lot, here's a named parking lot where you have the cars for the individuals have a name. And so now look at DNA as having lots of regions that have named parking lots. Where a molecular machine, only specific molecular machines are allowed to occupy that parking lot. This is going to change your perception of how DNA works, particularly in eukaryotes. This is very, very much a eukaryotic thing. Some archaea, which are prokaryotes, have some aspects of this, but it's really well represented and very sophisticated in a eukaryote. So I'm going to talk about what we call chromatin. The way DNA is put together in a eukaryote is through something called chromatin. So we have these, you see these little spheres here? Okay, so you see this DNA here?
Mm-hmm.
And the DNA is wound around these little spools or these spheres called histones. So you see the little sphere here?
Mm-hmm.
And so you can see the DNA wind, just like you have threads that are wound around a spool. DNA has, in eukaryotes, has this thing called histones. So the way that DNA is packaged, so you can either have like free-forming thread or thread that's wound around a spool. Again, these are weak analogies. In bacteria, they're kind of just free floating. In eukaryotes, they have to be packaged around these spools called histones. And so you have the word histone here. And when you have a histone and DNA wrapped around it, we call it a nucleosome. And they don't have the word here, nucleosome. So we have then these spools. And when you have these spools and the DNA wraps around, you have nucleosomes. And so this complex of nucleosomes is then called chromatin. And so you see the word chromatin.
Okay.
And so the chromatin then is organized into the chromosome.
Wow.
So just try to, if you have to repeat it, because that's a lot to just throw at you all at once. So just think about that. So, and the thing is, the histones have these little tails here. And that's going to be very important because they're like random access memory, if you're familiar with it. They're like little places for Post-its where you can kind of put little markers and signs, temporary signs, Post-it. There was a word that I had.
Post-it notes.
Yeah, like a Post-it note. Or a marker or a flag. Now, DNA itself can also have little Post-it notes that we call methylation marks. So collectively, we can call these things epigenetic modifications. So going back to Waddington, he's the one who came up. There he is, the guy. Oh, I just want to look like him. That's so cool. Waddington was the one that came up with the term epigenetics. And so the idea is you have the DNA that provides blueprints for proteins, but then you can both modify the DNA a little bit chemically by adding a little Post-it called a methyl group. And then you can also modify the tails of these histones with little Post-it notes. And those are called epigenetic marks. I prefer the word chromatin modifications. That's a little more accurate. So, and that's kind of the chemistry of this. We'll skip it. But let's look at an embryo after it got fertilized. This is a mouse embryo.
Okay.
So 3 hours after fertilization, we were able to highlight the changes in these epigenetic marks. So this on the top is the dad's DNA, and on the bottom is the mom's DNA. At 3 hours, they have all these epigenetic marks. So going back here to all the little Post-it notes, you can have Post-it notes here. In this case, what we're seeing are the Post-it notes right on the DNA. After 3 hours, they have all these Post-it notes. After 6 hours, like that. Then suddenly at 8 hours, all the Post-it notes are removed from the dad's DNA. Amazing. Orchestrated. And yet the mom is all lit. And then some hours later, it starts to split and they mingle. And then you could just see all the lights changing. It's just so cool. Let me show a little more what happens with the Post-it notes. This is in, let me see. You're actually helping me present this better. Okay, so we have like the mom and dad. And I don't know which one's the mother or father, but you could see like the two blobs of DNA. This is just beautiful, the process of conception. And then when they join, they start to split. The embryo starts to split. And you could see all the different colors of lights. So each of these little blobs is a new cell line. And each of the cell line has different patterns of these Post-it notes. The orchestration of this is what makes eukaryotic architecture just so spectacular. You don't see this. So now the cells start to differentiate, and you could see all the different lighting up a color, all the different epigenetic marks. And this is going to be a new living creature. This is the process of going from conception to development and birth. And just, you can just, okay, to tell you the complexity of this, just imagine you have a genome that's billions of bases, and you have to control all these Post-it notes in just like a symphony orchestra. and it has to be perfectly orchestrated. And what you're seeing here, what might look just kind of like trivial changes in, it's like, oh yeah, it's color. No, just think of billions of nucleotides being controlled in these switches. They have to identify the right nucleotide, the what do they call the cytosine, phosphate, guanine pairs. It has to identify that, and it's going to flip it. It has to flip the right ones. It has to know whether it's the mother or father. It has to be on the right chromosome. It has to be on the right gene location, etc. And then
wait, how is it flipping? Like, what is the process? Like, what is actually selecting or causing this to happen?
Machines. And I actually, that is such a beautiful question. I went to the professor when I was studying chromatin modifications at the NIH, and I said, how does it know where to flip it, when, and how? He said, we know the machines, but we actually don't know where that knowledge comes from. Where in the cell does it impart to the machines to do this? So we've isolated the machines that do the flipping of those states. We know, we have names for them. They're proteins and they're protein complexes, and I'll cover some of those here. But how does the timing actually work? Where collectively is all that knowledge to do this? This is beautiful, but then going back to the mouse example, going back to the mouse example. I'm sorry. Oh yeah. How does it know that, okay, at 8 hours, 3 hours, we leave it all lit up, but at 8 hours, the clock strikes and we shut all this down. Where's the information for that? We kind of know that, if we knock this or that out, that affects the clocking, but we only have pieces of the puzzle. And the professor said, we don't know. And I was like, yeah, this is a lot more complex, because I was asking the very same questions you asked. And this is so complicated, we don't have the answers right now. We're getting some of them, but I feel like we've only touched the tip of the iceberg of how all this is exquisitely orchestrated, and it has to be just right. In the Drosophila, this process, this differentiation process that I showed here, William Bialek pointed out, as you're seeing all this develop, each of these new cells, how they subdivide, it's within 1 minute of precision. If you're off, the creature's going to probably have a birth defect. 1 minute of precision. And so all these clocks have to be synchronized within 1 minute, and they're cross signaling. It's just amazing. And he said the amount of chemical signaling is just staggering. It could be the difference between one molecule difference between here and here will determine the fate of that cell line. One molecule. It's able to sense one molecule. And he was saying, he gave this lecture on biophysics. He said, more perfect than we imagine. He said, it seems biology, the way it does its craft is optimized to the limits of physics. You cannot have one smaller unit than one molecule to do signaling. And the bacteria, the embryos, not bacteria. The embryos are doing this. Their individual cell parts are timed within 1 minute, and sometimes as little as one molecule making the difference. How do you have machinery that can sense one molecule difference and then do all this switching? And I'm going to show you something really cool. This is a C. elegans here. You could see here, I'm going to just highlight. All right, so you see this little red thing here?
Yeah.
The little red.
Mm-hmm.
I'm going to give a zoomed in picture. There are the two reds here, and you can see the little blue marks. Mm-hmm. These are it's already having foresight. These are going to be the gametes. These are these are sex-related reproductive cells. And you see it's distinguished. It's all beautifully orchestrated. All the parts are there, and you can see all the computation that has to go on. I mean, this is mind-boggling that we have that level of computation. And that's why earlier you're actually helping me to kind of redo this slide. You ask such nice questions. You make me just excited to talk about this. I was facing a really hostile crowd when I was presenting this, and they were not happy about all this. You are, which I'm thrilled about. So there was a paper published in, I believe it was PLOS One. DNA is part of a DNA chromatin computer complex. DNA is part of a chromatin computer complex. She demonstrated that it's what they call Turing complete, which makes it a computer. So this whole thing where you see all these little tails and these little post-it notes, it's part of a big computation. So it has to go through this computation to make all those little lights flick off, on and off. And I'm just going to show a little bit of all the post-it notes. So let's go through, and then we'll try to wrap this up. So you have these little histone tails.
Mm-hmm.
The tails are actually locations you can put the post-it notes. So you have the histone. Here's another picture of the histone tail, and there's this tail here, right? So you have the chromosome. It makes chromatin. The chromatin, if you zoom in, it's made of DNA wrapped around these histones, and the histones have tails. That's just to give you a context there. Now we're going to zoom in on the tails here. The tails have all these positions where you could put all these molecular machines to dock and make modifications. This is, each of these locations is a single amino acid. Amino acid. And on this tail, you can put all these different post-it notes, and it ends up being like a like computer memory. And I'll point out a little later. Yeah, it's like computer memory. And I'm going to show a picture later. Look at all the different computer memory states that it can occupy. So when you're seeing all those lights flicker, you're just watching the computation, just the kind of in a global scale. But this is at a very molecular scale. All that just imagine all those lights flicking, and you could zoom in on what's happening. It'd take you lifetimes to see all the little, to account for all the little flickers that are going on in all the different regions. So you have machines. So you're asking, how does it do this? You have machines for the histones called readers, writers, and erasers. You have machines to do that. And I know one of our mutual acquaintances hates the term machine, and he would not like the idea of writing machines, erasing machine, and reading machines. But this is what's used in the literature. This is what's used in the literature: readers, writers, and erasers. And you asked specifically about, the machines that do this. It's a different set of machines that modify the methyl groups on the DNA versus the ones that do it on the histones. And I got this from the Stem Cells Handbook 2013. All of these processes they call it like epigenetic memory. It's called random access memory. And I'm going to show a picture of, I call this God-made random access memory. So I'm going to show you. So here's a picture in electron microscope of the chromatin. So this is the chromatin. The wire connecting the chromatin is the DNA, and then the DNA wraps around the histones there. And when you have DNA around a histone, it's called a nucleosome. So you see these little nucleosomes. Each of those are memory devices, as I was demonstrating. And I call this God-made random access memory. I want to show you what man-made random access memory looks like. This is the old magnetic core memory in our earlier computers.
It looks so similar.
I know. Okay, and then let's look to the abacus. This is kind of our first man-made memory unit, the abacus computer. And look at it here. I mean, I don't know. That just kind of warms my heart as an engineer. I just think this is just too cool. So now I'm going to show you how this thing can work. We have here, and I studied this, I had to do a term paper. One of these little markers here on the histone tail, on histone 3, the K is for lysine. Lysine has to get this little post-it note called either a methylation or acetylation. It's a methylation. And so you need machinery to do that. We have here the layout of our chromosomes.
Mm-hmm.
And on chromosome 12, there is a link RNA, a junk RNA, so they called hot air. It comes from chromosome 12. It somehow, and this is what it would look like, the DNA version. So you have this strand of the corresponding RNA called hot air. And there's a joke. They thought that this can't possibly be functional, so they'll just say, well, it just might be just a bunch of hot air. But they said, let's look at what it does. So that hot air, this RNA transcript is going to come out of chromosome 12 here. Somehow it's going to navigate. How it does that's kind of magical. Tough problem in biophysics. It's going to navigate from chromosome 12. It's going to go to chromosome 2. When it gets to chromosome 2, the hot air is going to find a parking lot in chromosome 2, specific out of the 50 million or so nucleotides. It's going to find the right location. Amazing. Pinpoint navigation.
Mm-hmm.
Out of the billions of spots that this thing could go, it goes to this one. Somehow it finds it. It finds it using let me see, I'm going to have to zoom out a little bit. It finds it using a bus called, let me see if I have it. Do I have it? It's not in this picture. Oh, it finds it using this thing called a SUSY 12 or a SUS 12 bus.
Mm-hmm.
So the bus will take the hot air. The hot air hops aboard the bus. Again, there are all sorts of codes with this. SUSY 12 will navigate from chromosome 12 to chromosome 2, find out, there are billions of places to go. Somehow it navigates. It has excellent GPS. It's going to find it, go to the right location on the DNA. On that DNA, then, it's going to assemble this complex machine that we call the polycomb repression complex 2. This is a massive machine, very complicated. And what it's going to do, you can see the chromatin here, the little wire, the black here are DNA wires.
Mm-hmm.
And there's this little histone tail, and it's going to grab that histone tail.
Oh.
Grab that histone tail, and let's go back to this diagram here. It's going to flip that one amino acid. This is like finding a needle in the haystack, but even worse. It finds it, it flicks it to a methylated state. What is the result of that change? When they were researching this, they said, in DNA that's above the waist, it's flicked one way. DNA below the waist, it's flicked another way. What they realized, the skin on your eyelid is very soft. The skin on the sole of your feet is much tougher. This little change is the difference. Look at all the machinery that has to be orchestrated just to get that little difference of a change. And this is only one small part of all the variety of cells. I'm going to stop sharing now. We can just...
And I feel like we kind of went from talking about eukaryotic cells and you took it out. But there's so much that we could have talked about.
Well, you see, this is what the eukaryote component has. That's why bacteria don't make multicellular organisms. They have the chromatin. They have the chromatin and histones and all that machinery. Today we
that's what's making it possible for
right. Mm-hmm. So what are eukaryotic components we've covered? We've talked about the nuclear import-export complex, all the shuttling. We've talked about the spliceosomes, and then now the chromatin. Chromatin gives the ability to do stuff. You don't find chromatin. You may get chromatin-like stuff in some prokaryotes, But the way chromatin is implemented in the eukaryote that's why I classify it as a eukaryotic component. You have another layer of computation that's just mind-boggling, that's expressed in eukaryotes, that's nowhere like that in prokaryotes. And that's why I say, how does that evolve? And if for nothing else, and when I was sharing this with Dr. Dan, he said, you know what, Sal, this is why you're highly respected, because you get into the nuts and bolts and you describe the complexity of biology, which is beautiful. And we actually had one moment in the debate, he said, that really is amazing. And I thoroughly agree with, this is an amazing complex. And so we had that one moment in this debate, basically over genetic entropy. This whole thing was in the discussion of genetic entropy. We had that one moment where we were just both of us were just sitting in wonder of life. And if, you know, this may have developed into the whole thing about examining evolution, but I feel like we could end on kind of an uplifting note and appreciate the wonder of life that's around us. And that just kind of ennobles, you know, we have a lot of mundane, terrible stuff happening in the world. But then I look at this, I said, there's something that transcends all this that's just glorious. And it's very comforting to look at.
And it seems like something that might require some foreknowledge or foresight, assembly, organization that goes beyond what was the alternative term you used for natural selection?
Differential reproductive abundance.
Something more than differential reproductive abundance could
which really
provide for.
Means, yeah, some creatures make more babies than others. That's all it really is. How can that explain? How can making more babies, making more offspring, how can you logically go from that to say it makes something like this? And boy, we're getting flooded with a bunch of, there must have been another show on, because now we're getting a lot of people joining examining evolution. We just blew through like a semester of molecular biology and a semester of chromatin modification, graduate school level, in an hour and 20 minutes. And Rebekah's still smiling after all this torture.
Yeah, it was fun. Go back and watch it, please. All right.
Would you like to highlight any particular comments before we
well,
close?
yeah, we can we
could at least
I'm
say
glad
hi.
to see
Hi, Danny.
Peter's here. Yep, good. And Jamie's here. Baby, I like Jamie's comment here. Baby steps and rainbows. I guess that's all we need for this process. And Dr. Joel has stayed around here, so that's I think he was going to go to bed, but he ended up sticking with us. So
yes, thank you for participating, Dr. Joel. I'm honored.
Yeah.
And then we have Danny here.
And Danny's here. I don't know if he's still here. He was here a little bit ago, but yeah. So, and I want to have Danny on to talk about natural selection sometime.
Did you know he and I are going to try to write a paper together on criticizing natural selection? I know someone's really going to be thrilled about that publication.
Yeah. Well, thank you so much, Sal. This has been wonderful. And thanks for everybody for being here. And good night.
Good night.